learning objectives. After the end of this lecture, you should be able to know the different pathways of each system, identify the nuclei involved, and understand how sensory receptors transduce information, and then locate and describe the associated cortices and their functions. So we'll begin with auditory, which of course is hearing. So when we hear sound waves, they're in fact made up of a variety of different components, but they are vibrations that take on this sinusoidal shape. Okay? So as I said, there's a couple different components of sound waves. The two that we're going to really focus on are frequency, which essentially is the pitch or the number of cycles of these sine waves. So if this was a second, this would be 2 cycles, as well as the intensity. So the intensity is peak to trough, which we interpret as loudness, which is measured in decibels, also considered amplitude. So the ear itself is going to be composed of a couple of different parts. The external ear contains the pinna and the auditory meatus, which essentially are going to kind of collect these sound waves and then transfer them to the tympanic membrane. So the tympanic membrane is this structure that starts the middle ear, and so it's composed, the middle ear itself is going to have the malleus, the incus, and the stapes. And so when these sound waves come in, they're going to vibrate the tympanic membrane, which in turn allows for these auditory ossicles to vibrate, which will allow for impedance matching. And so these guys, these auditory ossicles, synapse on what's called the oval window. And obviously there's air in both the external and middle ear, but when you get to the inner ear, there's going to be fluid, a couple of different fluids, but primarily perilymph. And so what happens is we need, for efficient transfer of energy, from the air to the fluid, and these auditory ossicles are what do that, their vibrations. The ability of them to vibrate, or their stiffness, is controlled by the tensor tympani muscle, as well as a couple other muscles that are located here, and that can allow us to better tune into different sounds or to dampen certain sounds, like the sound of your voice when you're talking. So as we move into the inner ear, there are a couple. Yep.
What's impedance matching?
So impedance matching is just that transfer of energy, right? So it's going to make sure that that energy has a high fidelity, so it's at least the same, if not amplified. Anything else? Okay. So with the inner ear, we've got another couple of components. We've got the semicircular canals. So we've got an anterior, a posterior, and then a horizontal. They end in what's called the ampulla, or cristae ampullaris. And then we've got the vestibule, which contains the utricle and the sacule, and they have what's known as the macula at their ends. And then we've got the cochlea, which we're going to talk about first. The cochlea contains the spiral organ, which we'll talk about in some detail. And just so you know, first order neurons that come out of the cochlea are in this spiral ganglia that's located here. So the cochlea itself, we took a cross section of it, is going to contain the scalae vestibuli, the scalae tympani, and then the scala media. So these two regions that outline or border the scala media are going to contain the perilymph I talked about, but these specialized areas that contain the sensory receptors are typically going to be filled with endolin. And there's an ion concentration between the two of these that allows for these neurons to be able to depolarize, and those ion concentrations are caused or generated by the stria vascularis. So within the scala media, we're going to have organocorti. It's going to be composed of inner hair cells and outer hair cells, which are going to be separated by the tunnel of corti, and they're going to sit on what's known as the basilar membrane. So if you take a look up here, the basilar membrane is going to be narrower at its base and wider at its apex. And essentially what happens is when these vibrations come in into this area, they're going to allow for this basilar membrane to move. Okay? And as it moves, it's going to correspond to particular frequencies. So it will move optimally at high frequencies towards the base, and that the apex will move optimally when you have lower frequencies. And so what that allows is for a tonotopic representation of different frequencies that's conveyed to the brain and the cortices that we'll talk about. All right, so you can understand a little bit better about how this transduction takes place. Here you can see the two different types of hair cells. And hair cells have these specialized projections called stereocilia. So the stereocilia are organized from shortest to longest, and they're connected together, and they touch the tectoral membrane. So when vibrations come in and they move the basilar membrane, these hair cells will also move and they'll bend on the tectoral membrane. So as they move towards their tallest point, it'll cause depolarization. So that concentration, the ion concentration gradient will allow for potassium to enter into the cell, and then it'll activate these calcium channels to open and allow for normal release of neurotransmitters to the cochlear efferents. Any questions about that? So we'll move into the different pathways. In your notes, there's one slide that's hidden, but it shows just kind of the different pathways, the different parts of the pathways that are laid out in a more simplified, like this is this structure that goes to this one and this one. But we'll look at it as it's laid out here throughout the brain. So the first one we're going to talk about is the monorow pathway. It's a little bit more simple. So you have input coming from the cochlea, projecting up over the superior olive as the posterior acoustic stria, and then that projects up through the lateral lemniscus to the inferior colliculus, and then from there to the medial geniculate, and then up to the primary auditory cortex. With the binaural pathways, it gets a little bit more complicated, and you're going to start to get into matching sounds between different ears, right? So that's known as coincidence detection, and that's how we preserve the tonotopic representation and how we localize sound. So what you're going to have here are input from the cochlea. It's going to come up and cross as a trapezoid body in the medial lemniscus and project to the superior olivary complex. Some fibers from the same ear, its lateral side, go to the same superior olivary complex. And the fibers that cross here in the trapezoid body, some of them will synapse here and then project to the superior olivary complex, and those are inhibitory information from the opposing ear. So either way, when you get to the medial superior olivary complex, this is going to be the first site where you get information from both ears. It's important for sound localization, and you'll see too that you still have that tonotopic representation, so low frequencies versus high. So from here, you're going to project up through the lateral lemniscus to the inferior colliculus. There are some neurons here that project to the opposite lateral lemniscus through this posterior tegmental commissure. As you project up to the inferior colliculus, you're still going to have tonotopic representation, but you're going to have a couple of different nuclei that are important. So you'll have the central nucleus, which is really about those frequencies and the tonotopic representation. You'll have the precentral and the external, or sorry, the pericentral and the external nucleus, and those are important for things like habituation. So when you constantly hear a repetitive sound, your ability to kind of ignore it after a while, as well as important in multisensory integration. And so from there, these fibers will then project through the pterygium of the inferior colliculus up to the medial geniculate nucleus. And this area also has a couple of different nuclei that are important. So you'll have the anterior nucleus, which again is important just for those frequency-specific areas, so tonotopic representation again. You have the posterior pericentral, and this one is going to project preferentially to the secondary auditory cortex, and that's again important for habituation, but it's also important for attention, so detecting novel stimuli. In addition to that, you'll have the magnocellular nuclei, and those are important for more multisensory functions, and they project to the association cortices. Okay, so you can see from there, it just project up to the cortices. Any questions about any of that? I know that's a lot of information. Great. So actually, before we go there, the other thing I want you to be aware of too, your book talks about it a little bit towards the end, but there's a descending pathway too, which essentially goes through all these same nuclei and projects back down to the cochlea. And what it does is it allows for modification of the basilar membrane, so how stiff it is, also the stiffness of those auditory ossicles, and it allows to change the ion concentration and the stiffness of the stereocilia. So it can dampen sounds, it can help you tune into sounds, just like we talked about before. So here are some of the locations of these nuclei. Here you can see we have the lateral lemniscus, we have the inferior colliculus, and then the commissure of the inferior colliculus for the top two cheeks. Up here you'll see the two different cochlear nuclei, so anterior and posterior. And then if we look here we can see the medial geniculate body here, and then we have the brachium of the inferior calypus, and the inferior calypus itself. Okay? So, moving on, we'll talk some about the different cortices. So the primary and secondary auditory association, auditory cortices are going to be located on the transverse gyri petal here. Area 41 is the primary auditory cortex, and area 42 is the secondary auditory cortex. The primary one is mainly involved in that tonotopic representation, and again, that secondary one is more involved with habituation, attention, and those sorts of things. When we get to a little bit higher order functions, we have the association cortices. So area 22, area 40, and 39. These are going to be comprised of Wernicke's area, which is important for speech reception. Okay, so I'll show you some videos that will kind of better explain what that means. And then in addition to these areas, we also have Broca's areas, which are important for speech production. So area 44 and area 45, which are the pars opicularis and pars triangularis. Another thing here is that these areas up here in Wernicke's areas, I forgot to mention, they're important for reading and writing. All right, so we'll move on. I have some interesting little videos to show you. So this is Wernicke's aphasia, and this is more so about speech comprehension. Okay, so
Look, good.
What are you doing today?
We stayed with the water over here at the moment, talk with the people, with them over there. They're diving for them at the moment. But they'll save in the moment. He'll have water very soon for him. With luck for him.
So we're on a cruise and we're about to
We
get to
will
June.
swim right here and they'll save their hands right there for them.
And what were we just doing with the iPad?
Right at the moment they don't show a darn thing.
With the iPad that we were doing. We, like, here?
I'd like my change for me and change hands for me. Who would happen? I would talk with Donna sometimes.
So you can see he has completely fluent speech, although there's not much sense to it, right? So he has impairments in comprehension. And he in fact had a stroke that occluded his middle cerebral artery and damaged those Wernicke's areas, or those areas associated with those regions. Oops. All right. So this young girl unfortunately also had a stroke that affected her middle cerebral artery, but it affected Broca's area. And so I'll show you some of what happened to her.
School and English class,
Okay.
and I book and I read it aloud, but I can't because strength, and so I done that, and also it's the same as the same kind of thing as you know.
Pins and needles.
Yeah,
like pins
the
and
same, and also farmers, but
leg.
yeah,
Leg.
yeah.
Good thing.
So you can see she has some difficulty with producing speech, but her ability to comprehend is intact. So that gives you a better understanding of those two association cortices and their functions. All right, so any questions about auditory before we move on? All right. So next we're going to talk about vestibular, which essentially is your orientation in space and your ability to coordinate different movement reflexes, particularly postural control. Right. So back to the structures of the inner ear. Now we're going to be talking about the semicircular canals as well as the vestibule. So you've got anterior, posterior, and horizontal. And remember they end in the ampulla. And then you're going to have the utricle and the saccule, which each contain a macula. Okay. So these areas are also going to have hair cells, except now they have this specialized little portion called the kinesiolum. And And they're still going to be oriented smallest to largest, and when they bend towards the penicillum, they will depolarize, okay? If the stereocilia bend away from the penicillum, they will hyperpolarize. So we have a little bit of a different structure going on here. So within the ampulla, you'll have what's known as the cupula, which is this kind of gelatinous covering that's over the hair cells. And so you can see there's not really anything like before in the organ porti where you had the tectoral membrane. There's not really anything here that's going to pull on them. This gelatinous substance is going to move as you move, and that's what's going to cause these hair cells to depolarize. And that's why they can go in either direction. Okay? The same thing happens here with the macula. They have an otolith membrane that covers the hair cells, and these otolith membranes contain otoconia, which have calcium carbonate crystals that kind of respond more so to gravity. Okay? All right, so those are the two things here. You'll notice too at the ends of the semicircular canals, you can see each of the ampullas and they have a different direction that they depolarize in. Okay? And then the same thing with the two macula. So in the saccule and in the utricle, you'll have what's called a streola, which is this little line here. And the orientation that the hair cells depolarize is shown by these arrows. So you can see between these guys, their directionality is covered completely. Right? Are there any questions about that? Okay. So just to kind of better illustrate how these hair cells work, what you can see here is just kind of resting state firing for these neurons. So here we have the ampulla, here we have the macula. And then what you'll notice here is if you turn to the left, then you will excite the neurons on the left side, right? They'll bend towards the penicillum. And if you, on the opposite side, what you'll see is an inhibition of those neurons. So they'll be hyperpolarized, right? Same, or the opposite thing, a similar mechanism for the opposite, turning the opposite way. And then the same with the otolith membrane in the macula. Okay? So it's all about the direction that they turn, and essentially what that allows is for these structures, these receptors, to act as comparator units, right? So they compare the input from both ears to better allow you to detect your movements and your spatial location, spatial orientation. Any questions about that? So we won't go over too much detail about these, but I'd like to point out a couple of things about the pathways here. I've described a lot of it in your notes for you, but just to briefly go over it. So you've got your different hair cells, and they're going to transduce information as first order neurons. They come in, and they'll synapse on the vestibular nucleon. So you're going to have a superior, a medial, or lateral, an inferior, and a medial. There's also the cell group Y, and the different pairs of semicircular canals, as well as the macula, saccule, and the utricle with the macula, they project differentially here, and I've noted that in your notes for you. These guys communicate to each other, so you've got commissural fibers, and then they will project to a wide variety of areas. So you'll have some that project up to the cerebellum. They do that through the juxtarestiform. These fibers come up with the juxtarestiform and then go into the cerebellum through that inferior cerebral peduncle. And then you'll also have some spinal fibers that go down the spinal cord. You'll have some that go to a vestibulo-ocular system, and then the vestibulo-cortical system as well. So just to kind of highlight some of these nuclei for you again, you can see here you have the medial vestibular nuclei, the lateral, which contain those juxtarestiform fibers, superior vestibular nuclei, and then the reticular formation, which is also important. And this is kind of the best one that I found in your book to show a good majority of ones. There are obviously others. All right, so vestibulo-ocular is in your notes. I put it there for you and outlined it, but for the sake of time, we'll kind of move past that. But with the vestibulospinal, I think this one's pretty neat. So you've got the lateral and inferior nuclei that project down to the spinal cord. They synapse on gamma and alpha motor neurons, and they preferentially will activate extensor muscles within your back to allow for postural control. And with these medial ones, they actually project the muscles into your neck, and they allow for coordinated head movements. And then with the vestibulothalamocortical system, these guys project up to the thalamus. They're going to go to the VPL. They're also going to go to the posterior nucleus and the anterior pulvinar. And then they'll project out to different cortical regions. So some of the areas involved are going to be 2B and 3A. These guys are important for the movement of your head, system movement of your head. Then you'll also have the premotor cortex as well as 7. These guys together will help you detect spatial orientation, your awareness of where you are in space. And then some of these guys actually project into the hippocampus as well, and I think this is really neat. So you'll have projections from the dorsal thalamus to the hippocampus and the vestibular nuclei. And what's cool is that these guys help to control spatial navigation. So for those of you who don't know, obviously the hippocampus is really important for learning memory, and Brett will talk about that in a couple of weeks. But the hippocampus has specialized cells called place cells, and those are what help you learn and navigate your way through space, right? So your ability to navigate through your house, your ability to drive to your class in the morning, that's controlled by or helped by these place cells in here. And so it's kind of neat because this vestibulothalamocortical system helps you with spatial navigation, or spatial navigation. That's pretty cool. Okay. So the last thing we will talk about today is olfaction. And olfaction, of course, is your ability to smell. So a lot of people don't really think a lot about olfaction because who really cares about your ability to smell? But it's really important historically, right? So our ability to find food, our ability to run away from predators, to find prey, all depends on olfaction. I'll tell you a brief story since we have some time. When I was doing my master's, when I was taking neuroscience during my master's program, our professor had us read this article about a experiment that went on. Experimenters had people exercise, so both members of the opposite sex exercise. They took their clothes and extracted their sweat, and then had people from the opposite sex smell the sweat and rate their attractiveness. So they apparently think that pheromones still exist in humans, although it's a really controversial idea. So it was really interesting. So smell is important for a lot of things, just to kind of highlight that point. All right, and so here's a little pathology for you. This is called an esthesioneuroblastoma. And so what happened here is this 38-year-old woman, she had a year-long episode of no smells and frequent headaches. Right? So she kept getting more and more headaches. She also had some nosebleeds, and then she started having some problems with vision. And what they found was that she had this gigantic tumor that started from her olfactory mucosa cells. Right? So eventually it completely destroyed her cribriform plate and disrupted cranial nerve 1, and it started to impede on her ability to see. So that's not that good, right? So the cells that kind of did that are going to be the mucosa cells that kind of underlie in this area. But this is essentially the structures you have before we get into the olfactory bulb. So here's that cribriform plate that it destroyed. And so you'll have the mucus layer down here that's generated in part by these supporting cells as well as this Bowman gland. You'll have supporting cells here that help secrete the mucus, but also help bind odorants to these olfactory receptor neurons. We'll talk about that binding a little bit more in detail. And then you're also going to have these basal cells, which are actually stem cells that will replenish these olfactory receptor neurons. Any questions about that? Great. So these olfactory receptor neurons, they have specialized receptors that detect odorants. Typically, they'll have one specific type of receptor, okay, or a receptor for a specific type of odorant, I should say. And they project to only one glomeruli, okay? So you're not going to project to multiple ones, just a single glomeruli. And what's been found is that these glomeruli, they actually will have synapses for olfactory receptors that contain a similar odorant receptor. So they'll respond to a similar type of odorant. Does that make sense? And I have some pictures that'll better highlight that for you in a second. Anyone have any questions about that? So the glomeruli obviously are in the glomeruli layer. They also consist of the periglomerular cells that crosstalk between the glomeruli and also send inhibitory connections to the mitral and tuft cells. They essentially help with the communication of all these cells together. The external plexiform layer is where you're going to have the cell bodies of the tuft cells, where you'll have the cell bodies of the mitral cell layers, and this is the mitral cell. And then with the granule cell layer, you'll have the granule cell bodies. Right? And so the granule cell bodies are similar to these periglomerular cells. They are the inner neuron of the olfactory bulb, and they form inhibitory feedback loops. Okay. So those mitral and tuft cells are the primary projections out into the olfactory tract, and they're those first order neurons. Okay. So the olfactory receptor neurons, as I was talking about before, have specific receptors for specific odorants. Right? So odorants come in, and it's not going to be just a straight depolarization like we normally think of. These go through a signal transduction cascade. So you'll have odorants bind to the odorant receptor, and then they'll activate they'll activate G-coupled proteins that will then activate either cyclic AMP or IP3, which will then in turn activate ion channels for depolarization. Yes.
Why are there different paths to work?
It's a good question. I'm not entirely sure about that. But we could talk about more. Any other questions? All right. And then we have the different cortical projections that they go to. So they'll project bilaterally to the opposing olfactory tract and olfactory bulb. And then they'll also project to different cortical regions. So there's a summary of these cortical regions here. So you can see they project to the contralateral olfactory bulb, but they also project to the piriform cortex, the amygdala, as well as the paramygdala cortex and the interrhinal cortex with the hippocampus. So you notice a lot of these areas are associated with limbic structures, so they're really important in learning and memory and those types of behaviors. And in fact, some species navigate entirely or a large portion of their activity is based on olfaction, particularly rats. I don't know how many of you work with them, but almost their entire brain consists of their olfactory bulbs. It's a huge portion of it. Any questions on that? All right. So a final pathology to talk about. So a while back, there was this guy named George Gershwin, who was a composer or conductor, and he was 38 when he started having headaches. Right? In addition to his headaches, he started having these weird hallucinations, olfactory hallucinations, so sensing a burning smell when there was none. So people thought he was crazy and sent him for psychotherapy. Right? And then a couple months later, he was seen for a doctor because he was having papilloedema. And essentially what that is swelling of the octave disc. And so a couple days after that, he went into a coma and then died. And then they found that he actually had a large glioblastoma in his right temporal lobe. So the moral of the story is that if you start smelling weird things, you should probably get that checked out. Our sense of smell is really important, and because these areas project to hippocampus and things like that, they're really involved in these temporal lobe structures. So when you have damage to them, or you have things like temporal lobe epilepsy, you can start to get these weird sensations, right? A lot of times too with temporal lobe epilepsy, you'll also have taste hallucinations, often with like metallic taste. So essentially what you need to know is the different structures and functions of the external, middle, and inner ear. You also are going to need to be able to describe each of those sensory receptors we talked about, and you'll need to understand the mechanism of how they transduce information. Okay? You should be able to locate and trace the pathways of each of these systems, and then know their location and function of their related cortices. And then you should understand the different pathological complications that arise when there's damage to any of these regions. Are there any questions?